Detectors in HPLC. HPLC analysis requires a detector to monitor the compounds themselves in order to quantify and identify the compounds separated in the column. In HPLC, the detector monitors the mobile phase passing out of the column, which further releases electrical signals directly proportional to the characteristics of the solute or the mobile phase. The detectors used in HPLC are as follows: UV visible absorbance detector, photodiode array detector, fluorescence detector, refractive index detector, evaporative light scattering detector, electrical conductivity detector, electrochemical detector, and mass spectrometer. Ultraviolet or visible spectroscopic detector. It measures the ability of solutes to absorb light at particular wavelengths in the ultraviolet or visible wavelength range. When the light of a certain wavelength is directed at a flow cell, the substance inside the flow cell absorbs the light, and as a result, the intensity of the light that leaves the flow cell is less than that of the light that enters it. An absorbance detector measures the extent to which the light intensity decreases, that is, the absorbance. There are three common types of UV visible absorbance detectors: fixed wavelength detectors, variable wavelength detectors, and photodiode array detectors. In fixed wavelength detectors, absorbance of only one given wavelength is monitored by the system at all times, usually at 254 nanometer. It is simplest and cheapest of the UV visible detectors, limited in flexibility and limited in types of compounds that can be monitored. While in variable wavelength detector, a single wavelength is monitored at any given time, but any wavelength in a wide spectral range can be selected. Here wavelengths vary from 190 to 900 nanometer. It is more expensive and requires more advanced optics, more versatile and used for wider range of compounds, and more sensitive due to photomultiplier tube or amplification. Second is photodiode array detector. It operates by simultaneously monitoring absorbance of solutes at several different wavelengths. Light from the broad emission source such as a deuterium lamp is collimated by an achromatic lens system so that the total light passes through the detector cell onto a holographic grating. In this way, the sample is subjected to light of all wavelengths generated by the lamp. The dispersed light from the grating is allowed to fall on a diode array. The array may contain many hundreds of diodes, and the output from each diode is regularly sampled by a computer and stored on a hard disk. Its advantages are determination of correct wavelengths in one run. After all peaks have been detected, the maximum absorbance wavelength for each peak can be determined, and a PDA detector can collect spectra of each peak and calculate the absorbance maximum. It can analyze peak purity by comparing spectra within a peak and scan spectrum very quickly, provides single beam, and is useful for kinetic studies. It is also useful for qualitative and quantitative determination of the components exiting from a liquid chromatographic column. While its disadvantages are large noise because the amount of light is small. Third is fluorescence detectors. Fluorescence detectors are the most selective, sensitive, and specific than all other HPLC detectors. Specific wavelength is used to excite and then emit light signal in analyte atoms. The intensity of light is monitored continuously to quantify the analyte concentration. Fluorescence is 10 to 1000 times sensitive than UV detector for strong UV absorbing components. Even a single analyte in the cell can be detected by the fluorescence detectors. For some compounds which do not have fluorescence absorbance or low absorbance, they can be treated with fluorescence derivatives such as dancyl chloride. The system is easy to operate and relatively stable. As we can see from the diagram, the light from lamp like xenon lamp passes through an excitation filter which provides essentially monochromatic light of the desired wavelength for excitation of sample molecules. This light passes through the flow cell causing sample molecules to fluorescence at a higher wavelength than that used for excitation. The resultant light passes onto the detector for quantification of the emission signal. As the fluorescent emission is very low, photomultiplier rather than photodiode is used. There are three types of fluorescence detectors: single wavelength, multi wavelength, and laser induced fluorescence detectors. In single wavelength detector, the wavelength of excitation is fixed and they are made from low pressure mercury lamps. In multi-wavelength fluorescence detector, it contains two monochromators to select wavelength of excitation. And in LIFTs, it shows optical emission from molecules that have excited to higher energy levels by absorption of electromagnetic radiations. It is used as separating tool for polymerase chain reaction. Determination of solutes like protein, nucleic acid, polycyclic aromatics, hydrocarbons, and toxic elements like cyanide. The fourth one is refractive index detectors. They are also one of the bulk property detectors and are based on the change of refractive index of the eluent from the column with respect to pure mobile phase. There are different types of refractive index detectors: Christiansen effect detector, interferometer detector, thermal lens detector, and dielectric constant detector. They are mostly used for detection of non-ionic compounds that neither fluoresce nor absorb in the UV region. They face the drawback of being less sensitive and need of temperature control and less suitability to gradient illusion. Here, the light emitted from the source is concentrated into the cell containing the sample and reference sample. Both the chambers of cells are separated by a diagonal glass sheet. The light passes through the cell and reaches the beam splitter that diverts the light towards two photocells. A change in the observed refractive index of the sample results in the difference in their relative output. In this, difference is amplified and recorded. Fifth one is the evaporative light scattering detector. Here, detection is based on the scattering of a beam of light by particles of compound remaining after evaporation of the mobile phase. It is a universal detector and does not require a compound to have a chromophore for detection. There are three steps involved in detection: nebulization, mobile phase evaporation, and detection. In nebulization, inside the nebulizer, the column effluent passes through a needle, mixes with the nitrogen gas, and forms a dispersion of droplets. In mobile phase evaporation, the droplets pass through a heated drift tube where the mobile phase evaporates, leaving a fine mist of dried sample particles in solvent vapor. In detection, the sample particles pass through a flow cell where they are hit with a laser light beam. Light scattered by the sample particles is detected, generating an electrical signal. The flow from the column is nebulized with a stream of inert gas. The mobile phase, which must be volatile, is evaporated, leaving tiny particles of analytes. The particles are passed through a laser beam and they scatter the laser light. The scattered light is measured at right angles to the laser beam by a photodiode detector. Sixth one is electrical conductivity detector. It is used in analytical applications of ion exchange chromatography for the detection of ionic compounds. Detector measures the ability of the mobile phase to conduct a current when placed in a flow cell between two electrodes. Conductivity detector measures electronic resistance and measured value is directly proportional to the concentration of the ions present in the solution. Seventh one is electrochemical detectors. It is based on the measurement of the current resulting from an oxidation or reduction reaction of the analyte at a suitable electrode. The level of current is directly proportional to the analyte concentration. Three electrodes are employed, which are working electrode, auxiliary electrode, and reference electrode. Electrolysis of analyte occurs at working electrode and auxiliary electrode supplies the current. Reference electrode measures the solution potential. Potentiostat is used to control the voltage at auxiliary electrode in order to maintain the constant potential difference between the solution and working electrode. Sensitivity is high and low detection limits can be achieved using this detector. The last one is mass spectrometer detector. Liquid chromatography mass spectroscopy, that is LCMS or HPLCMS, is an analytical chemistry technique that combines the physical separation capabilities of liquid chromatography with the mass analysis capabilities of mass spectroscopy. It is a method that combines separation power of HPLC with detection power of mass spectrometry. Mass spectrometry is a powerful analytical tool that can supply both structural information about compounds and quantitative data relating to mass. For using mass spectroscopy, there is a need for an interface that will eliminate the solvent and generate gas phase ions for mass spectroscopy. It has three distinct parts: ion source, analyzer, and detector. For all mass spectroscopy techniques, an analyte is first ionized in the ion source since the mass spectrometer can only detect charged species. Based on their mass to charge ratio, the ions are separated and focused in the mass analyzer.